Hi, now let's discuss about gene cloning and recombinant DNA technology, which has revolutionized our biology and biomedical engineering. So what is recombinant DNA technology? So it's often used in genetic engineering. This describes a set of techniques to transfer genetic information from one organism to another by isolating and amplifying DNA sequences for a particular gene. So this has a history of this recombinant DNA has revolution. So it starts with a study from bacteria. And because bacteria has a lot of invasion from virus, it has developed a mechanism to protect itself. And that has led us to actually engineer DNAs. So the discovery of 1960s, we learned how to cleave DNA in defined region. We now know how to cut the DNA in a specific region. And then 1970s, so that we cut the DNA, then that specific DNA sequence, we learned how to transfer this one into the bacteria. Then that DNA of our interest, it could be human DNA, and that can be amplified in bacteria, such that using bacteria as a factory for creating a useful proteins for human. So it's like a movie editing. So we have now material, genetic material as DNA, and in a specific region, if we have a specific scissors to cut and then edit it and then rejoin it and to create something that's useful. It's almost similar to the traditional classical movie making, because you take many films, and some places unnecessary you cut it out, some places you actually cut it in, put in, and you join them together. So this led to the ability to even engineer organisms. So for example, majority of the beans we are eating now are actually coming from genetically modified organism. It's called GMO. And in biomedical research, to study a specific function of a gene, we sometimes modify the gene, and so that we can understand the disease process which is relevant to that specific gene. So disease models of animals, we are actually using engineering these DNA. So on the other hand, we can produce therapeutic proteins and vaccines by using this recombinant DNA technology, such as diabetes. There's a type 1 diabetes, which is previously called as juvenile onset, because these patients cannot produce proper insulin. So it needs to have insulin from outside. In the past, they use sometimes a pig-derived insulin, which can cause an immune reaction and problem. Now, by this recombinant DNA technology, we can produce human insulin produced in the bacteria by using this technology. So for this, we have to discuss about cloning. So we've heard about cloning a lot. What does the clone mean? Clone means exact copy. So the creation of genetically identical copies of genes, cells, and organisms is called cloning. And what is molecular cloning? We are talking about a segment of specific DNA out of a genome, and we identify those and then cutting that segment and amplify them. This is what we call molecular cloning. And the gene of interest, it can be inserted into a cloning vector and using some kind of enzymes to open the plasmid at known locations. And later we'll talk. So bacteria has developed an enzyme which will recognize bacteriophage, which is a virus which parasites bacteria. So bacteria developed this enzyme to cut this exogenous virus, and such that to restrict the growth of this bacteriophage. So people found this enzyme and named it as restriction enzyme to restrict the growth of bacteriophage inside the bacterium. So to manipulate DNA, there are two enabling techniques which has involved. So first is the capacity to cut and change and join DNA segments together. And the second is a vector system for copying and mass manufacture the part of the DNA we are interested in, which is being carried by DNA. We call it as a vector. So a vector is a carrying vehicle that can hold an isolated DNA sequence of our interest. And it also holds replication, origin of replication, and selection markers together. So in a nutshell, a vector system which contains a foreign DNA, we call it as a recombinant DNA. And the output product of protein which comes from this foreign DNA, we call it as a recombinant protein. And this molecular cloning, once we have these, and we can amplify that recombinant DNA by using polymerase chain reaction. So the vector is a carrier to carry a specific gene of DNA of interest. And the most popular vector is actually a plasmid. And this plasmid usually is a small circular DNA molecules that can replicate independently of chromosomal DNA in bacteria. So that we call plasmid. And I want to show you how human insulin was made as a factory of bacteria by using this recombinant DNA technology, using this nice animation. So recombinant DNA is a technology that science developed to insert a human gene into the genetic material of a common bacterium. That the recombinant microorganism now can produce protein encoded by the human gene, in this case, human insulin. So this is so fast that I'm going to stop it here. So this is a human insulin gene, and there's a loop of bacterial DNA which is inside the bacterium. So now, by inserting human insulin gene of this DNA segment into a plasmid vector, which is a loop of bacterial DNA, and we create this. So now this is recombinant DNA, which carries a foreign DNA, and this is a plasmid. And we can throw it into bacteria that it independently as a DNA plasmid inside the bacterium, so recombinant bacterium. And if we make them to grow in fermentation tank, to grow and express its gene of human insulin, so it will produce multiple copies of insulin protein, and we will be able to harvest those insulin and purify it, become our medicine of we can inject into this human insulin into the diabetic patient. So let me explain a little of different terminologies for this. So in bacteria, the process of inserting a plasmid into bacterial cells by using microinjection or electroporation, and this we call as transformation. So transformation sometimes used for some normal cells becoming into a tumor cell. Now in animal cells, the introduction of a plasmid into a cell by using chemical or mechanical means, we call it as a transfection. But in other case, introduction of plasmid into a cell, when the method of the introduction is not chemical, not mechanical, but viral method, we use a little different terminology called transduction. So when you're reading papers, you may find out these subtle differences of inserting plasmid into a cell. So let's get into a little more, let's design a plasmid so for our own purpose. So very interesting, you see this piece of DNA, which is double-stranded, 5' to 3' end, 3' to 5'. You can see these. You can see, let's say, this sequence of interesting sequence of 6 nucleotide GAA-TTC that corresponds to CTTAAG. Interesting, one way GAA-TTC, you can read the other GAA-TTC. This is called a palindrome. And EcoR1 is a restriction enzyme, which when it finds to this sequence, this will cut out this way. So makes these 2 pieces of DNA. And this cleavage is done by EcoR1. And this weird name of restriction enzyme is coming from, we found it from E. coli and R, the first one. And this is a sticky end. And of course, there are enzymes which cut like just right through in the middle of defined sequence, which will create a blunt end. But for this recombinant DNA, sticky ends are very useful. So this is a typical plasmid. You can actually buy it and you can insert your own interesting DNA into that for your molecular cloning. So let's take a look. So this plasmid usually contains origin of DNA replication, and there is a tetracycline and ampicillin resistance. This is antibiotic resistance for selection. And there are a number of these restriction enzyme binding cleavage sites. So these restriction enzymes target, you can say targeted molecular scissors. And it comes from microorganisms such as bacteria's natural defense mechanism to cut up or destroy genes from invaders, such as virus that enter their cytoplasm. And these enzymes usually cut symmetrically like this. Although the symmetric cut can leave overhanging single-stranded tails. So this is overhanging on the either 3' ends or 5' ends. And this has a specific sequence which can attach specifically a complementary sequence. So we call it as a sticky ends. And these sticky ends are useful in inserting a gene of interest and reclose plasmid by DNA. This is called hybridization, complementary hydrogen bonding. So let's summarize. This plasmid is a small circular DNA molecule that can replicate independently of chromosomal DNA in bacteria. And this is popular vector for gene cloning, and it provides this resistance, antibiotic resistance. The reason we have this antibiotic resistance is when we put the plasmid into the number of bacterium, it's hard to know which bacteria uptake this. So by having this antibiotic, you give the antibiotic, and those who did not uptake this plasmid will die. And so those survived ones were likely to have this recombinant DNA. So this is molecular cloning procedure by use of a plasmid. So let's take a look. So the plasmid I just described, and there's a restriction site, and there's an ampicillin resistance gene, and LACT gene is another way of showing pigment color so that we can distinguish those who uptake and who hasn't. And we have this foreign DNA of our interest to insert. So we cut this one like corresponding sticky ends, and we cut this with a restriction enzyme such as EcoR1, And then we mix them together. What happens? And this will hybridize and insert that pieces of foreign DNA into this DNA vector, so plasmid vector. And then you put it into bacteria who may uptake or may not. However, we have this ampicillin resistance. So you put ampicillin antibiotic, then those who did not have this resistance will die, and only those who has uptaken this plasmid will have this resistance, so which will be survived and as this way. So the cloning procedure is number one, we cut and paste with the restriction enzymes and DNA ligase to connect them together. And we need that vector of DNA fragment that can replicate inside the bacteria, and putting into the bacteria is called transformation. So reintroduce human DNA vector into a bacterial cells. And then we can select those to find those bacteria containing our own recombinant DNA.